The overall goal of this protocol is to produce uniform aggregates of mouse embryonic stem cells that undergo self-organization and axial elongation in suspension culture.
This method can help answer key questions in the field of developmental biology, such as the role of tissue interactions in axis specification and self-organization in processes like gastrulation. The main advantage of this technique is that it allows developmental events to be studied from a novel, deconstructed perspective outside of the embryo.
Prior to aggregation, maintain mouse embryonic cells, or MESCs, in ES-LIF medium on gelatin-coated 25 square centimeter tissue culture-treated flasks. For aggregate generation, start with a tissue culture flask containing cells at 40 to 60% confluence. Aspirate the medium and rinse the flask twice with PBS. Add 1 milliliter of 0.25% trypsin EDTA and incubate at 37 degrees Celsius for approximately 5 minutes to detach the cells from the flask. Next, add 5 milliliters of ES-LIF medium to the detached cell suspension and mix thoroughly. Transfer the cells to a 50 milliliter centrifuge tube and take out a 1 milliliter aliquot to count the cells. Add 5 times 10 to the 4th cells to 5 milliliters of warm PBS and centrifuge the cells at 170 times G for 5 minutes. Aspirate the solution without disturbing the pellet. Then, add